is the last of our COVID CRB victims. So Erick started his thesis journey in 2019, fall of 2019. And so really was just getting his feet wet, as others in his cohort of experience, when all hell broke loose. And coming from a lab where we have a very tight peer group that where they rely on one another to teach each other techniques and take care of the animals together, being stuck in a room for 6 months where you didn't really know anybody, it was really tough. And so that was the way things started for Erick. And he was, with the other White Side members, incredibly generous with his time in getting all of our stuff here back, including animals. And I was thinking, let's brag a little bit. I think the White Sled had to move the largest animal. So I'm talking live animals, okay? Not like dead stuffed things. I'm talking live animals. They were the biggest. They were in water. It was hard. So all those guys did great, including Erick. He came, so I would say that I would describe Erick's time at OSU as full of resiliency, was able to roll with many punches that were delivered to him and has come out very strong. So he was interested in the energetics of symbiosis. So he studied Cnidaria animal symbiosis. He was very interested in energetics. And one of the things that I like to do in the lab is to have the students grapple with what exactly kind of projects that they want to engage in. And of course, this can be difficult when you're something by yourself for 6 months. But he was interested in energetics and how the partners exchange nutrients, which is sort of the centerpiece of Cnidaria animal symbiosis. But he wanted to couple that with our lab-wide interest in the different life history stages of our sea anemones, adults, asexual buds, which you'll talk about a lot at larvae. And so his thesis at the end really became a study of the intersection of nutrition, symbiosis, and life history stage of our little mouse anemone. So he's going to talk to you about this today. Erick is really the nicest human. He's a gentle soul. I'm speaking of his mother. He is a really gentle soul. He was the heart. He is the heart of the lab. Never an unkind word came out of this kid's mouth. The undergrads loved him because of that. They bonded with him. He had lots of students working under him, and so that gentle kindness is going to serve you very well. So with that, his title is Energetic Provisioning of the Essential Offspring in Response to Nutrition and Symbiosis State in Exaptasia diagonal. And I will say this is public defense. Once he finishes speaking, everyone except the committee can ask some questions, and then we will adjourn to another room and grill it.
Yeah.
Okay.
All right. Well, thank you all for coming. Again, I'm Erick, and thank you all for coming to my defense talk today. So a quick outline of how this talk will go is I'll start off with some background information into the Cnidarian algal symbiosis, and then I'll proceed into my 3 dissertation chapters and the experiments I conducted there, and then round it off with some general conclusions from my thesis. So to start off, just generally, what is symbiosis? We generally categorize symbiosis as an instance when 2 or more organisms of different species are engaged in a long-term association with each other. And we can subdivide that into different categories based on how each partner benefits from that interaction. So you might have a parasitism where one partner benefits and the other's harmed, such as in the case where a tick is attached to a human. We might have commensalism, where one partner benefits and the other is unharmed or unaffected, such as where we might have some barnacles attached to the skin of a whale. Or, as we'll kind of talk about today with corals, it could be a mutualism, where both partners benefit. So when we break down the Cnidarian dinoflagellate symbiosis, we have our, in this instance, a coral, which belongs to the phylum Cnidaria, but there are also other species, and that can engage in this symbiosis, such as the sea anemones that I'll talk about later, and some jellyfish. And what we can see here is like along all these branches, it's very brightly colored, very healthy and alive. But if we zoom in, we can see that this is not just like stony skeleton. It's actually made up of many polyps that like pepper the surface of the coral. And what's special about the coral symbiosis is that it's an endosymbiosis, meaning the symbiotic partner actually lives inside the tissue of these polyps. So this is where we see our dinoflagellate symbiont that is from the family of Symbiodiniaceae. So these algae make up many parts of like the cellular tissue like inside of the coral. And reason why these corals are so thriving in what would otherwise be nutrient poor waters is due to this nutrient exchange that happens between both symbiotic partners. So for instance, like the coral host will supply the symbiont with access to nutrients in the forms of inorganic carbon and nitrogen, as well as providing access to sunlight. The tissue itself is clear, and the algae pigment actually is what we see as the color on the coral. And so with access to carbon and nitrogen and sunlight, they have what they need for photosynthesis. And the coral host also provides a stable habitat for the algae to live in. And in return, the algae supply the host with nutrients in the forms of sugars, fats, and amino acids to make up proteins. So this exchange of nutrients is really the basis of the coral reef ecosystem and allows it to grow and expand and thrive. And with such a vibrant ecosystem, they provide a lot of benefits to the surrounding ecosystem as well as to the coastal communities that they're found in, such as being a hotspot for biodiversity. Roughly like 25% of marine species can be found like on or near corals. And because of that, there's lots of tourism that happens. Everyone wants to go diving on the corals and like see everything that's there. But they also provide some storm protection where we might have like some waves from like storms that might crest over the reef and will actually like lessen their strength and reduce the amount of flooding damage and that might hit like coastal communities. And in addition, they're big, they are the larval home for many different types of fish species. So they act as like the home and serves as nurseries for these large scale fisheries and for people to come fish on them. So they total like generate like trillions of dollars of revenue like every year. Um, and but although these systems like are pretty well founded and stable, they can also be pretty fragile in that symbiosis can break down pretty easily. So we have a healthy coral shown on the left here, but if changes to conditions such as like heat, like if we're having rising ocean temperatures, differences in lighting, whether there's like too much light or high light intensity or even not enough light, or in case where nutrients like in the surrounding water might be depleted, this can put stress on the system that leads to the loss of symbionts from the coral tissue. And this is like what we see as coral bleaching. The like I mentioned before, the pigment of the algae kind of provides a lot of the color that we see on corals. But then if the symbionts are lost, what we're left with is the clear coral tissue and the calcium carbonate skeleton, which is white colored. So because these reef systems are at such a risk for stress, it's important for us to understand the cellular and molecular underpinnings that make up symbiosis so we can learn how to mitigate possible future damage. And one of the ways that the Weiss lab looks at that is through this sea anemone called Exaptasia diaphana, which dang, I spelled wrong there, but commonly known as Aptasia. We use this as a model system to study the coral symbiosis. And a model system is where we have like a particular species like Drosophila or mice that we can easily use in a lab and study different systems. So why do we use it to study corals? Well, and for one, they're symbiotic with the same family of algae as in corals from the family Symbiodiniaceae. Uh, they are also, we can keep them as aposymbiotic, which means without symbionts. So we can see that on the left here. We can see that we just have the clear tissue and maybe like some pink brine shrimp leftovers, but for everything else, the host is healthy, it's tall, it has all its tentacles. So we can use this as a comparison to see like how the symbiosis is actually changing the host on a cellular and molecular level. Um, Otherwise, another reason we use Aptasia is because we can keep them in a lab much more easily than corals. Corals are very particular about their environmental conditions, so they don't always do well in lab settings. But with Aptasia, we can keep them in aquarium tanks and or even like little plastic tubs with seawater, and they do great. And we can generate these clonal lines. That means they can be asexually propagated very easily, which is what I'll talk about a bit today. But this allows us to replenish our animal stocks like very easily, so we can have enough animals to use for some high throughput experiments. So, as Virginia mentioned, I got really interested into the life history of Aptasia. So on this diagram, we kind of show both aspects of the sexual and asexual life history strategies of Aptasia. So we have, for one example, we have like our adults in the middle here. And from the sexual side, if we rear them up to be mature enough and large enough, they can actually release gametes, and about once a month now that we have that set up in our lab. And they can form these planula larvae that subsist on like lipid droplets that will fuel their needs for development and growth until they can acquire symbionts from the surrounding water. And so they are born, these sexual larvae are born without symbionts, so they need to acquire them first. But once they do, they can reestablish that symbiosis and start exchanging nutrients, then proceed into becoming juveniles and adults. I talked a lot about the pedolacerates, which are the asexual offspring of Aptasia. So what happens here is like if we look at like the base of the Aptasia here, that's what we call the pedal disc. So from this pedal disc, the Aptasia will crawl across the surface and pinch off a piece of its pedal disc that forms what we call the pedolacerate. And interestingly, the algal symbionts are already present like in these lacerates. So we already have a possibility for early establishment of symbiosis, which we'll get into kind of the effects of that in this talk. But once they form and like fully heal themselves from that pinching process, they can start their progression through their different stages of development to juveniles and adults. But we don't necessarily know a lot of the nutrient dynamics that occur during this stage as we do with adults and larvae. So that's what I focus on in this talk today. And it's important for us to understand how these nutrient dynamics work at asexual life stages, because one of the main ways that we engage in recovery of reefs or like propagation of reefs is through coral gardening, which is a method of where we can break off pieces of healthy coral and they can like regrow themselves and like form clones with that parent, but it allows us to generate more and more pieces of corals and populate these nurseries until we can maybe reestablish them in new locations or in their previous homes. So to start off, my first chapter looks at the effects of heterotrophy and symbiosis on metabolite provisioning in pedolacerates. So we've talked a bit about different ways that they can acquire energy. So we like to call Aptasia a mixotroph because it has multiple sources that it can acquire nutrients during its lifetime. One of those being an autotrophic method, which is where the algal symbionts supply nutrients, as I mentioned, in the forms of sugars, fats, and proteins that they can transfer to the host and meet like most of its metabolic requirements in order to live. And then the other method is through heterotrophy, which is where they can predate, engage in predation on small marine organisms such as like brine shrimp and other small marine organisms. And the nutrients that they can acquire from these processes allows them to fuel their metabolic needs to engage in homeostasis, so maintaining its regular body functions, allowing for enough energy to grow, and then engage in reproduction. So I'm primarily focusing on the aspects of growth and reproduction for this chapter. So what I wanted to ask with this experiment is like if we had an adult and we put it through different types of feeding regimens, like first off, how would this affect how the adult is behaving? Would it be growing, shrinking? Would it have other things happening? And then if we took that, like how would those like test conditions impact like what is present in the pedalacerates in terms of their total energy reserves? So how we went about this is we took both symbiotic and aposymbiotic animals, and we split them up into about 8 different test conditions based on whether they were kept in 12 hours of light, 12 hours of darkness, so that they had access to light for photosynthesis to have that autotrophic food source, or if we kept them in total darkness, so where we eliminate that factor and possibly have the algal symbionts just sitting there and not doing what they normally would. And then whether we fed them 3 times a week with brine shrimp so that they would have that heterotrophic food source, or if we starved them completely. And we maintained them in these test conditions for about a month. And during the beginning and end of this time period, we measured the same individuals, the same adults, their pedal disc size as a proxy for growth rate and see like whether they were growing or shrinking. And from there, we cut pedal lacerates from the pedal disc so that we could generate lots of individuals for us to pool together and have multiple replicates for our tests. And then we would grind these up to release all the symbionts and metabolites that would be present in the tissues. And for a separate experiment, we took the same test conditions, but from the lacerates, we kept them intact for a test where we stained specifically neutral or energy-rich type lipids with this dye called oil red O, which I'll talk about later. But from this, when we ground them up, we pelleted everything out so that we would pellet the symbionts, but leaving the metabolites like in the suspension. And we divided that up into 3 different assays so that we can measure total amounts of carbohydrates, lipids, and protein to get a full sense of their energetic reserves. And finally, we quantified symbiont density using a cell counter to see like if symbiont densities were affected by the nutrient regimens that we put them in. So the first set of results I'll show is specifically looking at the changes in pedal disc area. So this is the overall change from day 31 and day 0. So whether they had a net increase or a net decrease. And then on the x-axis, we'll have like our feeding conditions, and the colors represent whether they were symbiotic or not, or whether they were kept in the light or dark. So for this first group, where I've abbreviated things to APO for aposymbiotic and SYM for symbiotic. And just a quick note, when I show these different letters above my results, this is indicating the specific pairwise comparisons that I did between test groups to see whether there were specific differences between each group. So for instance, here, what we can see is that feeding had a major effect on growth in aposymbiotic adults, where we can see those that were fed had a net increase in growth, like after 1 month, and those that were starved had an overall net decrease over 1 month. And we can see the non-shared letters between these two sets of groups indicates that there was a statistically significant difference between them. But interestingly, when we looked at our symbiotic animals, what we can see is that most of our groups that were either missing one or both nutrient sources had relatively similar or no changes in pedal disc size or even a decrease over time, and they were not statistically significant between each other. Only our symbiotic fed group that was in the light had an overall increase in pedal disc size after about 1 month. And this is interesting because it is appearing that if 1 or 2 sources of nutrition is missing from these adults during this time, that can possibly lead to it either just maintaining their regular body size or resulting in a decrease. So there could be possibly a cost of symbiosis occurring during this time when there's not sufficient nutrients present. So overall, we can see that feeding leads to an increase in size in apo adults, and that SYM adults apparently need both access to feeding and lighting in order to grow. Next, I looked at the levels of total carbohydrates in our lacerates across all of our test conditions. And interestingly, what we could found is that we needed, again, all of these sources. We needed feeding, we needed lighting, and we needed symbiotic state to result in abundance in total carbohydrates. Whereas like all of our other test groups did not statistically differ between each other. So this might be suggesting that carbohydrates might be used up very quickly as a source of quick energy, and that only the combination of all of our treatment effects leads to a substantial increase in carbohydrates that they can have stored up for later use. So it seems that we needed all three of our variables in order to show an effect on carbohydrates. Next, I looked at amounts of total protein, and what we can see is a pretty linear relationship going from our symbiotic fed groups in the light all the way down to our aposymbiotic starved groups by the end. So we have major effects of feeding and symbiotic state, like on the amounts of total protein that is present, where our symbiotic fed groups tend to have the highest amount of protein possible, and those that were starved and aposymbiotic having the lowest amount of protein possible, which makes sense. We're having heterotrophic food sources. They can be getting sources of nitrogen and other things to fuel protein synthesis, and our aposymbiotic ones that are starved, they're not getting sources from any kind of symbiont, and they're lacking heterotrophic food sources. They tend to be the smallest ones present. So we can see that symbiosis and heterotrophy led to higher levels of protein in our lacerates. Next, I looked at our total lipids that were present. And when I looked at our symbiotic lacerates, we could see that there was no major differences between any of our test groups, whether it was based on feeding or lighting. But interestingly, when we looked at our aposymbiotic test groups, we saw a large increase in the amount of total lipids present compared to our aposymbiotic, or compared to our symbiotic test groups. So we have this major effect of symbiotic state on total lipids, which is interesting because we would think that with symbiotes being present, there would be transfers of fats and things to supply other nutrients to the host. But previous work from a former Weiss Lab student, Trevor Tibby, gives us some insight into this. So he found that there might be more structural lipids present, which adds into our total lipids because he found that there was high cellular turnover in aposymbiotic individuals and more cells undergoing replication and a higher density of cells. So it's possible that aposymbiotic individuals might have higher amounts of total lipids to replenish all the cells that they're going through during this time. And so it seemed like that apos overall had this higher concentration of total lipids. But then we're going to compare this to our oil red O study, which, as I mentioned before, is this lipophilic dye that stains neutral and energy-rich lipids, so very ones that are used for storage and used for energy specifically. And this dye is a red color stain, so the more red that is present is indicating the more energy-rich lipids that are present. But you can see a difference compared to our total lipids results. We can see our symbiotic individuals having much more uniform red staining across all of its tissues compared to our aposymbiotic individuals that have pretty low levels, so like pink in this fed group and practically none in our starved group here. So it seems that Simlacerates had a greater amount of this oil red O staining. And when we quantified this, what we found is a large effect of symbiotic state on this oil red O concentration, where our symbiotic test groups were statistically different compared to our aposymbiotic individuals. So in this case, what we're seeing is that symbiotic lacerates had a greater concentration of energy-rich lipids specifically. So to kind of wrap up this chapter, some of the major conclusions that we found is like if we had this adult, our growth in symbiotic individuals is primarily affected by having both autotrophy and heterotrophy present, whereas apos only really needed heterotrophy in order to grow. And when we looked at energy reserves, we found that Carbohydrates were specifically affected by the combination of all three of our variables here, and needed all three of them in order to generate a high abundance of carbohydrates. And our lipids were primarily affected by symbiosis. So whether we were looking at total lipids or energy-rich lipids, that affected which group had higher concentrations than the other. And our protein was primarily affected by our factors of symbiosis and heterotrophy. So this moves us into my next chapter, where I wanted to examine these effects of feeding it on energy reserves and symbiotic growth, but in developing pielasters. So taking it from that one snapshot point, like right when they're formed, and now extending it out into a time series. And a lot of work in the Weiss Lab has looked at pielaster development already. One of our former postdocs, Jason Presno, he really characterized the step-by-step process that pielasterates undergo as they develop. So he found that the early stages of pielaster development are engaged where we have that initial site of detachment from the adults, and that wound site closes up, and from there the tissue starts to invaginate and close in on itself and form this new oral disc where a new mouth will form, and also where we can see these tentacle buds start to sprout out because all the tentacles like form away from the oral disc. And then these later stages of development are characterized based on their nutrient sources. So in our aposymbiotic test groups, they're solely relying on the stored nutrients that might be present in mesenteries, which is what Jason hypothesized would be the case. And they can continue to grow as long as they have a source of nutrition, but otherwise they might start to arrest cell growth or just stop at a certain point, and they'll proceed later once they have more food. Whereas symbiotic lacerates have to reestablish symbiosis every time that we have new cells forming, and then algal replication to move into these new cells as they're progressing into the body column, into the oral disc, and into the tentacles. But once that happens, there can be a switch from the stored nutrients, which is primarily lipids, and then we can re-engage in that transfer of photosynthates from the algae to the host. And one of our other postdocs, Sam Bedgood, he took this timeline that I decided to replicate from his experiment. He tracked pielaster development over a course of 60 plus days, and he used mean tentacle number as a proxy for growth. And we can see from his data, our aposymbiotics animals are shown in black. Our inoculated lacerates, which were aposymbiotic originally, but he inoculated them with symbionts later. Those are in like pink, purple color, and symbiotic individuals are in blue. So he found that there were three main periods of development through lacerates. This first stage where those that were inoculated or aposymbiotic started to grow a little bit quicker during these early stages compared to symbiotic animals, possibly with the reestablishment of symbiosis happening, resulting in a slower development rate. But as we proceed into period 2 and period 3, the symbiotic individuals started to grow much faster at a higher rate, with inoculated individuals falling close behind, with apos kind of being the last ones here. But in this case, these were all individuals that were being fed, which are why these are in closed circles. Those that were starved had a different relationship where if they were engaging in symbiosis but were starved over time, they actually saw this large decrease in body size and growth until we reached little tissue balls, or they were completely dead at this point. So whereas apo individuals were able to maintain a relatively stable growth for a period of time. And this kind of goes back to the idea I was mentioning that symbiosis might be incurring a cost on the host if like nutrient sources aren't present. And this could be with a possible increase in symbiotic density. He found individuals that were symbiotic but that were starved had a large increase in symbiotic density by the end of the experiment. So this gets into the idea that the coral symbiosis might not be as mutualistic as we think. It's a very fragile thing, as I mentioned, and if there's stress on the system, the symbionts might actually end up hoarding nutrients for themselves and not transferring them to the host. There's a breakdown in the control of nutrient exchange, and the symbionts will proceed to replicate, but the host ends up suffering if that occurs. So I wanted to replicate this experiment and see like whether this was specifically a result of like energy reserves being depleted and looking at how that might occur. So I wanted to look at energy reserves and algal growth over time based on our effects of symbiosis, feeding, and then I added in this effect of life stage where I repeated this experiment in adults to see whether this is a life stage effect or is this mostly a nutrient-based effect. So what we had is we had our adults that we cut lacerates from at the beginning, and we generated enough to set up 4 different time points that corresponded to the 3 periods that Sam observed in his work. So we had one right from the beginning, and then one about a week after when the wound site should have closed up. This is when a mouth can start to form, we can start feeding them, but this is before they really proceed into those intermediate or middle stages, which I have represented by the stage 21 time point, and then one time point by the late stage, so roughly a month after treatment began. So we generated enough lacerates for adults to split up into groups where we either fed them rotifers 3 times a week or we starved them completely. And we used rotifers in this experiment as opposed to brine shrimp in the previous chapter because these lacerates during this time don't have a large enough mouth to feed on brine shrimp. So rotifers are actually small enough that they can ingest them completely and get a heterotrophic food source. And so when we looked at our results between our lacerates and our adults, we took some measurements, again, looking at pedal disc area to determine how growth is changing over time, as well as our total energy reserve. And then we also added in some measures to look at symbiont health with mitotic index, which is an indication of cellular replication, as well as a measure called FV/FM, which is analyzing photochemical efficiency and like how healthy the algae are and how well they're engaging in symbiosis. And for this talk today, I'm mostly going to focus on the mitotic index and the total energy reserves data. So when we we'll look at our adults first. So when we look at our total amounts of carbohydrates here over time, we didn't see too many effects like between actual test groups, but we did see an effect of the age or like how many days they were in the treatment. So we can see that groups started to level off and like had higher amounts of carbohydrates by the end of the experiment than they did at the beginning. And when we looked at lacerates, we saw a similar effect where we didn't see many differences among our time points and our test groups, but we did see an overall increase in these carbohydrates by the end of the experiment, with also an effect of symbiotic state, where both of our symbiotic test groups here, kind of colors are shown at the bottom. They had higher amounts of carbohydrates than their aposymbiotic counterparts by the end. So we can see that there appears to be an effect of time on both our adults and lacerates with some effect of symbiotic state. And in our lacerates, this could be relating to Jason's theory that as symbiosis progresses and as they start to age, they might be relying more on photosynthate transfer. So we're seeing a higher amount of carbohydrates being present at the end of the experiment compared to the beginning. And when we looked at lipids, we saw in adults a similar effect of what was happening in the previous chapter in terms of total lipids, where our aposymbiotic test groups had higher amounts of higher concentrations compared to our symbiotic counterparts here. While again, we still didn't see many pairwise comparison differences between our time points and test conditions. And with our lacerates, what we could see is that we again saw this effect of time where our levels of total lipids like change dramatically like between each time point. And we did see a little bit of an effect on feeding where our starved individuals had more than our fed individuals. I think this also kind of goes back into Jason's previous idea of the switch between stored nutrients and photosynthetic nutrients where they could be relying on these lipid stores like during like these early stages, but then we might see a decrease in the uses of these compounds or their abundance, and then that switch into carbohydrate levels instead. So overall, we see that this total lipid concentration is changing based on symbiotic state in our adults here, and then based on feeding and time in our lacerates. And lastly, when we looked at mitotic indexing, so this measure of cellular replication among our symbionts, in our adults, we didn't see any differences detected among our sample groups. This could be indicating that symbiosis is pretty well established in these individuals, and there might not be dramatic changes in like how symbiont replication is being regulated by the host during this time, or it might not be happening within the time scale of this experiment. And then when we looked at lacerates, we did see an effect of days post laceration or age on our mitotic index. Though this is primarily driven like by the end of the experiment, where we start to see symbiont replication increasing a lot by the end. But again, we didn't see a ton of differences between our test groups at each time point. But this could be because now that a symbiosis is becoming more established in the host, that we might be seeing like larger amounts of algal replication to start populating the new tissue and really building up symbiont populations for photosynthate production. So we can see that primarily our effect in lacerates is this mitotic index changing over time, whereas in adults we do not see much of an effect. So overall, from this chapter's experiments, we could not confirm like our previous findings from Sam's data. So we could not adequately determine like whether it's specifically an energy reserves issue that could be resulting in the data that he saw. But we did see that in our symbionts, these energy reserves and symbiont proliferation is a little bit more dynamic in lacerates compared to our adults. Adults seem like pretty stable for most of measures that we saw here, whereas in lacerates we saw the lines going up and down all over the place. So it does seem a little bit more dynamic. There might be more going on, and because it's like newly formed, it might be trying to have a lot of things becoming reestablished and put back together. And interestingly, unlike in Sam's data, we didn't see that mitotic index was primarily affected by feeding in this case. But this, all of this talk with like lipids really got me interested in characterizing the lipidome of aptasia and really understanding like what is happening in terms of those compounds, because we're seeing different relationships based on our symbiotic states between our life stages. So I wanted to explore more of these lipid compounds in my last chapter that I'll talk about today. So we can talk about studying different cellular components in an animal. So if we have our example of our aptasia here, we have different levels that we can examine compounds based on like whether we want to look at like specific changes in like genes or like gene upregulation, downregulation, whether we're seeing differences in like protein levels in the animal, or specifically like what I look at was the metabolomics portion of it. So if we're looking at the different classes of lipid compounds that we might be seeing and their abundances. And for lipidomics, we might have things such as like phospholipids that are making up the membrane, glycerides and glycolipids that can be used for energy sources, sterols and fatty acids as well, which are some major lipid compound classes that we see here. And with this, we can like generate a complete profile from an animal. So if we have like our different classes of compounds kind of shown on this y-axis here, we can use this field of lipidomics to determine like their relative abundance in a sample. So whether they're more present or less present, and whether that changes based on different test conditions here. And lipidomics and aptasia has been pretty well studied so far in our adults and in our larvae. But again, we don't know as much about how this changes in pedolacerates. So what I wanted to do here is I wanted to take our factors of symbiotic state and then our three different life stages, and then see how this lipidomic profiles changes based on our effects of symbiotic state and our life stage. So how we went about this is when we had our reproductive anemones. After many failed attempts, they were finally able to spawn and we produce gametes, and we fertilize those gametes to generate our larvae. And on that same day, we cut a bunch of lacerates from adults that we would use so that roughly our lacerates and our larvae were the same age. About 3 and 4 days past fertilization, we inoculated our larvae with symbionts. So we did 2 rounds of inoculation to ensure that these larvae were indeed inoculated with symbionts and that they would be present, and that we would see changes happening based on the reestablishment of symbiosis here. And we kept a subset that was aposymbiotic. We did have to harvest these a little bit earlier because they were starting to look very slow and much smaller compared to their symbiotic counterparts. So there might be some effects of harvesting them a bit early, but we were able to still gather enough to sample and have enough replicates for testing. But by a week after, we took the rest of our adults, lacerates, and symbiotic larvae, and we ground them up again to retrieve a specific lipid fraction that we would use for what we call mass spectrometry. So how this works is we take our sample and we load it into this big machine, and as it flows through the machine, it gets retained on this column, and the compounds will come off this column based on their weight and the solution that's used. So you can draw things off the compounds at different times and use this information of mass to help you identify compounds later. And as they come off the column, they undergo this process called electrospray ionization, which is a technique that doesn't have to break the compound into fragments like other methods. It keeps it intact and instead adds either a positive charge or a negative charge to the compound. And we ran it in both modes, like either attaching a proton or an electron to the compound, because different compounds have different efficiency with which they take on ionic charges. So in this way, we were able to recuperate as many compounds as possible from all of our test groups. So we can use that effect of charge as well as mass to be able to more easily identify which metabolite is being detected on the machine and its relative intensity, so which relates to its abundance. So this first figure that I'm going to show is called a principal component analysis plot. This gives us a general overview of where our lipid profiles among our test groups line up with each other. So in triangles, we have our adults. In squares, we have our lacerates, and circles are our larvae. And then apo individuals are covered in blue, and symbiotic individuals are covered in red. So what we can see pretty clearly here is that this principal component 1 explains pretty much all the variants we were detecting amongst our sample groups, which is likely the effect of symbiosis. We see our sim adults and sim lacerates groups on the left side of the graph here, and all of our aposymbiotic individuals cluster kind of on this bottom right corner. So we can see that but we can also see that our lipid profiles for our lacerates were pretty clustered amongst themselves and not overlapping too much with either the adults or the larvae. So this is kind of indicating that our lacerates might be indeed its own unique life stage class that's not exactly related to adults here. And interestingly, we also saw that our symbiotic larvae were clustered more amongst our apo groups than its other symbiotic test groups. And this could be because within 1 week of being inoculated with symbionts, it might not have pushed these lipidomic profiles closer to our other symbiotic test groups. Instead, it might be more closely related to being aposymbiotic. But we still see that it is roughly clustered amongst itself, not exactly overlapping with each other here. This next plot that I'm showing is called an upset plot, which shows us the specific total amounts of compounds that were detected amongst our samples, as well as whether these compounds were shared or unique to our specific test groups. So we just look at this bottom left figure here. What we can see is total amount of compounds detected per. And the main thing that we can see here is that our symbiotic test groups at every life stage had more lipids that were detected compared to their aposymbiotic counterparts. So we can see this comparison between sim and apo larvae. Here we have our sims had more. The sim lacerates had more than the apo lacerates, and sim adults had much more than the apo adults here. And if we look at the larger figure here, which is showing our unique compounds and shared compounds between test groups, it means it's unique if it's just a single dot that's not connected to anything. But if the dots are connected, that means this amount of compounds was shared amongst those test groups. So this y-axis is showing the just total number, and that's seen here on the top of each bar figure here as well. And what we saw is that there was only 3 main groups that had unique compounds to them, primarily belonging to symbiotic adults here, which had about 26 compounds unique to itself. Apo adults only had about 4 that were unique to them, and we did see 1 lipid that was unique to symbiotic lacerates. But we can see that most of these compounds were shared amongst 1 or more test groups that we found between these different life stages. It seems that as sim anemones begin to develop and grow, they're producing unique lipids as compared to our aposymbiotic individuals, which don't seem as producing as much unique compounds. And then our lacerates and larvae seem pretty similar as well over time. This next figure I'm showing is a heat map, which is showing these relative abundances of each compound that we detected, which is each row on this figure. And these abundances were centered and meaned around a value of 0. And then the colors indicate like whether this abundances were higher than the average or lower than the average. So if it's in the red, that means it's in higher abundance, and if it's in blue, that means it's in lower abundance. So if we looked at going by each life stage, we look at our apos and our sims here, we see that in our adults, we're seeing much higher abundances of most compounds present compared to our aposymbiotic individuals, where we're seeing a lot of this yellow-green color indicating that it was centered right around 0 or centered around the average value. And when we looked at lacerates, we saw a similar effect, though maybe not as in the same intensity compared to the adults, where we're seeing lighter orange colors for many of the lipids here, but that was still in higher abundance compared to its aposymbiotic counterparts. And then in our larvae, we saw that most of these compounds in symbiotic larvae were centered around 0, though we were seeing some compounds that were in this light orange color, but apo individuals had a lot of this blue color, which is showing that a lot of these compounds were in lower abundances amongst our apo larval sample groups. So we're seeing that these relative abundances of lipids are changing across both our effects of symbiotic state and life stage, it seems. And now the last set of figures I'm going to show is I decided to focus on 2 main classes of lipids for this talk. And this figure is showing relative abundance of specific compounds. So this group is called ceramides, and each color represents a different compound that was detected. And these were all tested to be statistically different, either based on our effects of life stage or symbiosis. And the y-axis shows their relative abundance or their percentage that makes up the whole of the compounds that were shown here. And what we're seeing with ceramides is we're seeing a lot of higher abundances for different ceramide compounds. So we can see that if we're looking at maybe this orange color here, we're seeing those are they're taking a lot higher percentage compared to its aposymbiotic counterparts. And we can see that across several different these ceramide compounds detected here. But we can also see some effects of our life history here, where we can see they're not exactly the same across each life stage, whether that's adult, lacerates, or larvae. You can see that some of these colors are higher or in lower abundance based on that effect as well. And just to talk a little bit about why we might be seeing more of this happening in our symbiotic animals, is that ceramides are an important and intermediate compound in this sphingosine rheostat that we see, and mostly what we need to know is that ceramides can be converted into either this SPH or S1P compound that determines like whether a cell is going to undergo apoptosis or cell death, or if it's going to undergo cell survival and be maintained. And ceramides have also been shown to be very important in regulating like symbionts and whether they get digested or expelled from the host, or whether they're maintained or able to proliferate. So it makes sense that we're seeing a ton of different ceramide compounds in our symbiotic individuals because these seem very regulated in maintaining symbiosis. And lastly, I wanted to show a ton of these triglyceride compounds. There was like so many that we were able to detect among our test groups. So we have a lot of different abundances shown on this figure, but overall we can start to see that we are seeing like some changes in compounds based on like symbiotic state, and we can see also that some of these compounds are differing based on life stage. So if I'm looking at just this blue one here, we can see across adults, lacerates, and larvae, we're seeing a difference in abundance of like what they make up of total. And we can see that there are a ton of compounds present in our symbiotic individuals compared to our aposymbiotic individuals. And triglycerides are an important part of the photosynthetic pathway. So symbionts will be able to produce triglycerides and transfer to the host. And these can be stored as lipid droplets in the host tissue and used for later energetic needs. And when we cleave off this glycerol backbone from this fatty acid change, we get a ton of energy released from this process. So it makes sense that we might be seeing more of these triglyceride compounds in our symbiotic individuals because they're being produced from this photosynthetic pathway. And as we've seen that before, that the act of engaging in symbiosis does seem to require a lot of energy for it to be maintained. So some overall conclusions that we can draw from this chapter is that we're seeing these lipid profiles change based on symbiotic state and life stage. We are seeing that lacerates are forming their own distinct life stage group compared to adults and larvae. And we're seeing that symbiosis leads to an overall increase in abundance as well as like diversity of like different lipid compounds that were detected among the classes that we showed. All right, and to wrap up my talk today, to go over some general conclusions from this work, what we found is that autotrophy and heterotrophy were able to enhance growth rate in our adults as well as our metabolite abundance in our lacerates, and that we're seeing a possible cost to symbiosis when one or more of these energy sources is removed. We can see that this abundance of lipids specifically can vary based on not only our symbiotic state, whereas in apos, we notice a greater amount of total lipids, and symbiotic individuals, we notice more energy-rich lipids. And this could also be affected by the life stage that we're seeing. So we might have things that are more present in adults or lacerates or larvae. And lastly, what we can see is that lipids do represent a unique life stage in aptasia, and they can make for a very useful tool for high throughput experiments. They are a wonderful mechanism to use in the lab. You can generate lots of numbers for your experiments, and I think we're going to have a lot more of lacerate work coming out of the Weiss Lab very soon. So just to quickly give everyone acknowledgements, thank you so much to my committee, Virginia, most of all, for all of your support and through these years and going through everything with me. Thank you to Molly and Dee and Kim, who should be online. Thank you for your advice and wisdom and helping me through all the stages of this process. And thank you to my GCR, Kathy Magnuson, for keeping everything well put together. Really appreciate it. Thank you to everyone in the Weiss Lab, past and present members. Thank you for all of our adventures together out on the coast, our mahjong nights, just being great mentors and friends throughout this whole time. And I was able to get too many pictures, but I had some incredible undergrads that helped me through a lot of these experiments. I really couldn't have done it without them. Thank you to everyone in the IB office staff. Like y'all really keep everything running, and it really means a lot. Just everything that you do for us, it helps everything. So thank you so much. Thank you to all of my mentors in teaching, from Lindsay, Stacy, and Devin, and in the A&P faculty, as well as like Carmen and Lori from the by 22X series. Uh, all of y'all taught me like how to be a better teacher and mentor, and I think it really shows up in like how I've been able to work with undergrads during this time. Nate Kirk has been a valuable member of the Weiss Lab. I taught with him a little bit for the invertebrate bio class, and he's been a big member of the Weiss Lab for so many years, and he's been a great mentor as well. And Adam Schoenard, he really helped me get my footing in terms of like what it meant to be teaching and engaging in pedagogy. So I really appreciate all of his work. Thank you to like the greater IB community. We've had so much fun together over the years, and like I really feel like I'm part of a special group because we are a lot closer than a lot of other departments, and I really appreciate like how we're all there to support each other, especially my cohort who hope they're all listening now, so they're all off onto big and beautiful things, but I really appreciate them going through this journey with me. Thank you to all my amazing friends who've been there to support me, like from our drag race watching nights, our board game nights, D&D sessions. It really means a lot to know that I had a great community out here to support me through everything. It's hard to think that it all happened, but it did. I'm really blessed for it. Thank you to my amazing family. My mom is here today. My brothers and dad are supporting me from afar, so they no idea what I've been doing the last like almost 10 years, but they are just so supportive. They don't care. They show up, they listen, they send me clippings of things I might be interested in. But they've been great. And now I have some amazing nieces and sister-in-law to share that with too, as well as our great family dog, Bonnie, our scary pit bull. So scary, just the sweetest dog ever. And thank you so much to my partner, Jadarion, who's here in the audience today. He has really believed in me so much and has been putting dreams into my head. It's been amazing to have him here during this time. And we've had so much time together, and he's fallen in love with Oregon. So I might be sticking around. So we'll see what happens. And then of course, I can't end anything without thanking the floppiest, bestest bunny, Rami, aka Romulus. Like he's been through my entire adult life. I can't believe he's like 10 years old now, but he's really seen me through living in Atlanta to moving out here. He's been through it all, and it's been amazing to have him here. And now I'm ready to answer any questions.
He then gets a mention of acknowledgement.
How could I not?
Okay, so we have 1 minute for questions. So we have, we're a little short on time, but let's open it up to the audience for questions.
So a lot of this interpreting the abundance of these different metabolites and lipids and so on. So that's going to be a function of utilization, storage, also feeding rate, right? So how are you accounting for potential differences in feeding?
Oh, so for like specifically with like the last chapter.
I guess it applies to all of the chapters. Like,
Yeah, so with at least with the last chapter, things were kept unfed. So this is just specifically what was generated like at the moment of creation. So there's not as much of an effect with feeding there, but with the others, so what would can you repeat? Like
yeah, so like when you're like comparing relative abundances and so on, that's just like a snapshot of what's going on in the organism at that time, right?
yeah,
So we tend to interpret that in terms of utilization or storage. But those aren't the only immediate things that can shape it at a given moment, right?
right. So mostly, yeah, I don't have a good answer for how we were able to do that. So it's mostly like a snapshot of like what is going on during that time and like whether we were supplying them with enough heterotrophic feeding or access to sunlight. So can't exactly determine like how much they were using in a given day. It's more like an overall snapshot of like the trends over time.
Really nice job with your presentation, awesome work. Probably see this coming a mile away, but are there any applications of your work to help corals withstand bleaching events?
So there is a lot of work in the West Side that I know June is doing a lot. So we are studying the effects of how like heat intensity and bleaching is affecting asexual reproduction. So you'll have to tune into like his work later. But we are seeing like some effects of heat actually like increasing asexual development. So it's possible that this could actually be accelerating the process. But that will have to be determined later. So I don't know too much about that.
Andre,
With her work, she was showing a lot of effect with feeding being able to help with resilience in bleaching. It helping to prevent like the severity of bleaching over time. So as long as they had access to enough nutrients, I would say that they would have like a greater resilience. So as long as they have some access, I think it would be helpful.
From Zoom, Nate, great talk and work, Eric. For the differences you see in the ceramides are possible changes among treatment due to the algae synthesizing the products in the symbiotic samples.
Sorry, I don't think I quite heard.
Okay,
So, fine.
so for the differences you see in the ceramides are possible changes among treatment due to the algae synthesizing the products in the symbiotic samples.
It's possible. We weren't able to exactly separate factors of that. So we're really taking like an overall look at like what the profiles were looking like across all these life stages. We probably need further tests to examine like whether they were being utilized for specific methods or not.
Yeah, this from Mark Nova. Could you help me understand how in chapter 4, the first PCA axis explained an impressive 97% of the variation, despite there being so much seeming structure in which lipid groups were called the larvae versus adults / symbiotes versus asymbiotes?
Yeah, so it was surprising to see like that much of a difference between our samples by symbiosis. So we're still kind of in the early stages of like analyzing a lot of this data. That we got this about a month ago, and I was like really rushing to finish everything. So those effects might change based on other factors, but it seems like yeah, it seems like symbiosis was like primary driver of this, but it'll be just need to be determined later, like whether that's exactly the case or not.
Really great job, Eric. I especially loved your figures. They made it really easy to follow. I feel like after being here and listening to Weiss Lab talks for many years, I should know this, but how old do the individuals need to be before they can start reproducing?
So at least in terms of sexual reproduction, we have to get them to a very big size. So we're feeding them almost like every day in order to have that happen. It's been a lot of work to actually get them to sexually reproduce, but Olivia and Maria here were able to get that done. But yeah, they need to reach a certain maturity and body size in order to be able to like start producing like gonads for that. So we don't generally see a lot of that in some Aptasia labs because you have to do so much and to get that ready, and then they have to be cued onto a specific light cycle to kind of mimic the full moon for them to release gametes. So it's a pretty intense process, but they do have to be very large. So this took like a couple years for us to actually get them there.
So when you're looking at 30 some days and calling it late stage, that's nowhere near a reproductive size individual. So that wouldn't explain any of their metabolic resource usage.
No, I shouldn't.
all right, so before we clap, Kim, we'll send you a Zoom link. Yeah, can you tell me the? I don't have my.
Yeah, yeah. Yeah, well, I'll send you a Zoom link. I'm just gonna say it right. Or today, you can put it in the chat. But yeah, we'll send a Zoom link to your email. We'll get that set up in a
So
second.
we adjourned the turn to 4329, which is near my office on the fourth floor of Gordley. And thanks everybody for coming. That's kind of here.